Namaskar, welcome to PK's classes. Today we are going to study about the types of autophagy. So you can refer to our earlier class on autophagy. So autophagy is derived from two words. One is auto means self, and second is phagy means eating. So a process in which cell eats its own contents. So the organelle which can digest the cellular content, we know that is the lysosome. Lysosome present inside the cell, and it can eat or digest the cellular contents which are dysfunctional and which are unnecessary. So the unnecessary and dysfunctional cellular contents, the organelles, are digested or eaten up by lysosomes. That means when the cell wants to survive, the cell, suppose the blood supply is less, there is ischemic condition, ischemia, that is decreased blood supply, or there is hypoxia, decreased oxygen supply. So instead of dying, the cell wants to survive. In that case, it can eat up a part of its cellular contents. We can say, call them as the waste materials, the cytoplasmic waste materials, or we can call them these dysfunctional and unnecessary organelles as the cytoplasmic waste materials, or we can also call them as cargo. So these cargo will be eaten up by the lysosome, and the energy requirement of the cell will decrease, and the cell can survive in the adverse conditions like ischemia or hypoxia. There are basically three types of autophagy: macroautophagy, microautophagy, and chaperone mediated autophagy. So when the cargo is less in quantity, the lysosome inside the cell can digest. In that case, we call it as microautophagy. And if the content is more, then there will be a role of autophagosome, and we call it as macroautophagy. And when some proteins showing signals or motifs needed to be digested, then chaperone is used. For example, heat shock proteins are used. That is called chaperone mediated autophagy. So let us discuss one by one. Suppose macroautophagy. This is macroautophagy, where there is initially a formation of a membrane to which the organelles which are unnecessary and dysfunctional, they adhere. These are the cell organelles, and the proteins which are dysfunctional and unnecessary, they will adhere to the membrane which is formed, and that will form a vacuole, and there will be vacuolization of all these cellular debris, and we call ultimately as autophagosome, a double membrane vesicle called autophagosome, and that autophagosome will now combine with the lysosome, and the lysosome will release the enzymes into the digestive enzymes into the autophagosome, and they will digest the cellular debris present in the autophagosome. So ultimately that will cause the degradation of the cargo or cellular debris. And ultimately after digestion we will get the sugars, the amino acids, and the fatty acids, and these can be used up. They can be again reused. So this is macro autophagy. So when the cellular debris is more, the cellular debris has to be removed for the cell survival, then there will be initially a formation of a membrane to which the cellular debris will adhere, and ultimately a double layered or double membrane autophagosome will form, which will bind with the lysosome, fuse with the lysosome, and the lysosome will release its contents into the autophagosome. Now this combination is known as autolysosome, and then the digestive enzymes present in the lysosome will degrade all the cellular debris, and in this way it will extend the cell survival. In case of microautophagy, the lysosome present in the cell, they will, suppose this is the cell, and this is the cell, and there is, suppose this is the lysosome, and the cellular debris, suppose this much is there, and it will be eaten up by the lysosome itself. So in that case, so there will be no formation of autophagosome. The lysosome can phagocytose the cellular debris or the cargo, and decrease the unnecessary and dysfunctional cellular organelles, and this case only a small amount of cargo is digested, so it is called microautophagy. Then there is a role of heat shock proteins. And for example HSP70, so that is called chaperone mediated, and that occurs when there is requirement of digestion of the proteins, and those proteins will express on their surface some motifs which will be recognized by the heat shock proteins or chaperones, and that chaperone will now help in its translocation into the lysosome inside the cell. So in the role of we call it as chaperone mediated, because chaperone helps in translocation of the proteins to be digested into the lysosome. So that is called chaperone mediated autophagy. So you can see here the chaperone helps in transport of the protein into the lysosome. Now let us discuss the benefits of autophagy. And autophagy can prevent cancer by suppressing processes like DNA damage, chronic inflammation, and genome instability. Autophagy can enhance the muscle performance by balancing energy use within a cell. Then autophagy can prevent neurodegenerative diseases by properly removing the misfolded proteins inside the neurons. For example, in Alzheimer's disease, the amyloid beta protein accumulation occurs. So autophagy can clear them, remove them, and can prevent neurodegenerative diseases like Alzheimer's disease. Then autophagy regulates inflammation, can decrease inflammation within our body, and autophagy can reduce the effects of aging because autophagy along with intermittent fasting boosts production of human growth hormone, and in this way it can reduce the effects of aging. Then autophagy, another term called mitophagy, where the mitochondria undergoes an autophagy, and that enhances cellular energy, that favors the development of new and stronger mitochondria that can produce more cellular energy. So these are the benefits of autophagy. So this is all about the different types of autophagy. Thank you.